Good morning. I looked through most of your study guides, okay? Good stuff for the most part, but I do want to go over a couple of organelles that are important, especially for later on, what we're going to do in the next couple of chapters, right, when we talk about cell respiration and photosynthesis. Right, so we're going to cover the mitochondria and the chloroplast, and then we'll go into the cytoskeleton, okay, which you did not have to cover on your own. All right, but you covered elements of the endomembrane system, right, the endoplasmic reticulum, the Golgi apparatus, right, you covered the nucleus, etc. All right, so you are still responsible for those. Understood? Good. So, I don't know how long my voice is going to last, so we'll just go with it, all right? Mitochondria and chloroplasts, two organelles, and from those of you that already did this, where do they come from? What do they do? Well, I just said what they did, so you should be able to figure it out. What do they do? What does a mitochondria do? It makes energy for the cell, right? It makes energy. It makes ATP for the cell by breaking down some type of carbohydrate, okay? It utilizes oxygen as well, right? So that's an important part. And chloroplasts actually transform light energy into and store it as chemical energy in the form of glucose, in the form of sugar, all right? And chloroplasts are found in plants and algae, and they're the site of photosynthesis. One other important thing is a peroxisome. Peroxisome is like a vacuole, all right, an opening, a compartment within the cell that is important for oxidation, right? It's an oxidative organelle, which means it breaks down free oxygen species that would otherwise cause problems in the cell. Okay, so we'll go through those. So, but where do the mitochondria and chloroplasts come from? All right, some characteristics of these two. They are enclosed by a double membrane. All right, so they have their own double membrane. They have a membrane. Within themselves, they contain free ribosomes and DNA. What do you know about cells? Free living cells. What do all cells have to have? DNA. What else do all cells have to have? A membrane. What else do all cells have to have? Ribosomes. So that's three of the four things right there. Right? So that evidence suggests what? That's evidence that supports the fact that at some point, chloroplasts and mitochondria were free living cells on their own. Okay? Those couple things, right? And the fact that they grow and reproduce somewhat independently, right? That tells you that that's what we use for evidence and support what we call the endosymbiont theory of origin of mitochondria and chloroplasts. That at some point in time, right, they were free-living organisms, much like bacteria and photosynthetic bacteria that are present in the world today. They are very similar, okay? But at some point in time, a eukaryotic cell that ended up with a membrane-enveloped nucleus engulfed or brought in somehow a mitochondria, another bacterial cell that was free-living, and hey, I can offer you, there's a deal that was sparked. The bigger cell with the nucleus, the eukaryotic cell, said, I can offer you plenty of free space and plenty of energy, right? Sugars, And in return, you break that down and you offer me ATP, and we're going to be happy together. All right? And that's what's happening. That's your endo-symbiont theory. That an early ancestor of eukaryotic cells somehow engulfed first a non-photosynthetic prokaryotic cell, which formed an endo-symbiont relationship. What does endo-symbiont mean? What's endo? What does that mean? In or within. Okay? What's symbiosis? What's symbiosis? It's a relationship between two organisms, right, where they were going to interact together. So they're going to live together, right? One's going to live within the other one. That's what. So the mitochondria is an endosymbiont, meaning that it's going to live within this eukaryotic cell. So first, the mitochondria was engulfed, okay? And then second, somewhere down the line, okay, you get the merging, you end up with the mitochondria, and then at least one of these cells may have taken up a photosynthetic prokaryotic cell. Right? So first, there's a engulfing of the mitochondria that establishes eukaryotic cells, and then at least one of those cells engulfed a chloroplast and then give rise to photosynthetic photosynthetic eukaryotic cells, right, such as plants and other cells that contain chloroplasts. All right, so there's a progression there. What evidence do you know about plant cells that suggests that the mitochondria came first? What if it were opposite? What if the chloroplast came first? What if the chloroplasts were engulfed first? Do plant cells have mitochondria? Yes or no? Yes, they do. Okay? That's evidence to support that. Right there, the fact that plant cells have mitochondria is evidence to support that the mitochondrion was engulfed first. Because if the chloroplast came first and then they diverged somehow, then all eukaryotic cells would have chloroplasts and only some plant cells wouldn't have mitochondria, right? So think about it flip-wise. We know that plant cells, photosynthetic cells, have chloroplasts, but not all eukaryotic cells are photosynthetic. But all eukaryotic cells have mitochondria, right? Plants and animals, etc. So they have to come first. And this is the diagram that's showing this. Okay? So you have your ancestor of your eukaryotic cells. Note there's an endomembrane system. There's a nuclear envelope. There's no mitochondria yet. Right? So at this point, the cell is surviving on providing, generating ATP through glycolysis. Right? A cellular process that produces a very small amount of ATP. It engulfs the mitochondrion, it develops, it engulfs. Now the mitochondria produce large amounts of ATP through cellular respiration using oxygen, right, breaking things down and allowing those cells to work together. And at some point, the chloroplast then is engulfed. Right? That's your endo-symbiont theory. It gives explanation to why cells have mitochondria and chloroplasts. Questions on that? All right. So mitochondria, they're going to break down their structure. They are composed of a double membrane. Okay? They have a smooth outer membrane and an inner membrane folded into cristae. All right, so it kind of looks like this. Here's your smooth outer membrane, then you have an inner membrane that is folded. All right, so those folds are cristae. Right? The inner membrane can makes two compartments. The intermembrane space. So this space right in here, that's your intermembrane space. Okay? So there's different parts of this mitochondria. This is very important when we talk about cellular respiration. All right? When we talk about how the mitochondria produce energy, this is the place that we're going to look. We're going to know the inner membrane in blue, the outer membrane in black, and the space in between them is very important. Because the way it works is that, excuse me, they're going to, the mitochondria are going to move hydrogen ions into that intermembrane space and produce a concentration gradient. Okay? Lots of hydrogen ions in this space, and they want to go back, and they're going to use that in order to produce ATP. Okay? Okay? So, what you end up with is the intermembrane space and the mitochondrial matrix. The matrix is this part in the middle, right here. This is your matrix part, right? And then you have the intermembrane space. Some of the steps of cellular respiration are done across this. This is the important part. This inner membrane, some of it's going to happen on the matrix, within the inner membrane, and some of it's going to happen in the intramembrane space. Okay? The folding allows for a large surface area for the synthesis of ATP. Okay? So, what do we have to do? What is this in another example of in biology? Why are we talking about the structure of mitochondria? There's a relationship between structure and function. Okay? The structure of the double membrane, the increased surface area, allows for the maximal function for cellular respiration and the production of ATP. Okay? So this is what it looks like again, right? Diagram, mitochondria. Here's the free ribosomes and DNA. That's right. Very reminiscent of prokaryotic cells and bacteria, which you look like free floating already. Okay, and then this is the electron micrograph showing a mitochondria. Questions on that? Now, chloroplasts are somewhat similar in structure. Okay, they have a double membrane. Actually, they have a very complex membrane structure. Okay, they contain chlorophyll. All right, plenty of other enzymes and proteins that are important for photosynthesis. We know that chlorophyll, more like borophyll. No, chlorophyll is found in leaves, and chloroplasts are found in leaves and other green organs of plants and algae because they perform photosynthesis. Okay, the structure of these includes thylakoids, right? It's not just a simple double membrane like we saw in mitochondria. It's more complex, right? So the thylakoids are actually membranous sacs. So the kind of chunks or vacuoles or vesicles of membrane that contain chlorophyll. All right, those sacs are stacked like pancakes into what we call a granum. Okay, and then stroma is the internal fluid, right, where those sacs are, the stuff that's between all the thylakoids. Okay. Now the chloroplast is one group of organelles that include what we call plastids. Okay. So let's see what a chloroplast looks like. I'm not going to attempt to draw this one. All right. So like I said, a more complex structure. Double membrane, intermembrane space, same as other mitochondria, but within the matrix, where the matrix of a mitochondria would be, you have this stroma and these thylakoids, membranous sacs which have chlorophyll that allow for the capture of light energy. Okay? And you see in the electron micrograph over here that those dark kind of spots, dark rectangles, are actually the thylakoids that are stacked up. That's what a granum is, right? So these are little thylakoids that are stacked on top of one another, your pancake stacks, okay? And you also note that they are all linked. They're not really separate. They're all linked to one another, all right, which is important in and of itself, all right? And you see down here chloroplasts in red in the colorized electron micrograph to show you that it's very dispersed throughout the cell. Why is it important that they're dispersed, or they cover the entire cell of a photosynthetic cell? What was that again, Brett? To absorb as much light as possible. More surface area, more light, right? That allows for more efficient photosynthesis. Okay? Last little one is peroxisomes. All right, and this is important in oxidation. These are specialized vacuoles. Okay? You read about vacuoles, hopefully, okay, in the beginning of chapter 4. Okay? But peroxisomes have enzymes that produce hydrogen peroxide. Okay? And then they convert it to water. What's hydrogen peroxide? Is what? H2O2. Hydrogen peroxide. They convert it to water. Plus what? Oxygen. Plus oxygen, right? Have you heard of free reactive oxygen species? Have you heard of those? No. Have you heard about antioxidants? How many of you heard of antioxidants? What do they do? They help build up your immune system. Yes, kind of. They get rid of these free-moving oxygens. Okay? So these are important because they actually, oxidation occurs, right? Which is, oxidation is losing, right? Losing electrons. Electrons going to be lost somewhere. All right, and then they end up reacting with other stuff. If this is a reactive oxygen species, okay, it's not stable. It wants to react with something. But then when you have an antioxidant, it's something in your body that reacts with this and prevents it from reacting with other proteins that can be important. Okay? Like their job is to absorb these extra things, right, and prevent them from causing damage to other cellular structures. Okay, so here's a peroxisome kind of wedged in the middle there. Why is it important when we're talking about a mitochondria and a chloroplast that we talk about this? What are mitochondria and chloroplasts doing? They're helping to create what? Energy. Okay, and when you create energy in the way it's created, the process, it actually builds up a lot of these reactive oxygen species. So the peroxisome catches them and keeps them from causing trouble in the cell. Questions on peroxisomes, mitochondria, and or chloroplasts. Now, peroxisomes they're not in the endosymbiont type. Okay, they're really a vesicle with some specialized enzymes that are produced by the cell. They don't have a double membrane, they don't have DNA, they don't have free ribosomes. Okay, so they're not an ancestor or a descendant of some other type of prokaryotic cell. Okay? I did not have you cover in your study guides the cytoskeleton because it's a little more complex than I wanted you to go through. Most of you have probably covered the nucleus before, yes? Right? So same thing with the endoplasmic reticulum, and hopefully you understand the function of it as well. Right? So that's a lot more straightforward than the cytoskeleton. Okay? But the cytoskeleton. Cyto, anytime you see that suffix cyto, it refers to cell. So really, this is the cell skeleton. All right, and what does your skeleton do? Supports, protects, right? What do you think the cytoskeleton does? Supports and protects, right? We're not very original when we come to naming stuff. We function, name, pretty much, pretty close in terms of relationships. Okay, but it does organize cell structures, and it provides an anchoring point and a movement point for organelles. Okay, basically organelles move in the cell, yes? You agree with that? Well, how do they move? They just float around? No. Some of it's very directed. Okay? You'll see that in a little bit. But this is the cytoskeleton. Kind of a crazy looking structure. All right, and what you're looking at right there are two different, red and green, two different elements of the cytoskeleton. Okay, in red you have microfilaments, which is one aspect, one subunit of the entire cytoskeleton. All right, you'll see the red is kind of in different places, yes? Different places in the green. There are places where they come together, and you can kind of see that in yellow, where they co-localize. But the green is tubulin, microtubules. Okay? And they kind of look like a spider web originating from the middle out. All right, there's a microtubule organizing center that we'll talk about. And we'll talk about actin filaments as well. And there's another part of it that's not, another part of the cytoskeleton that's not labeled here, intermediate filaments. All right, so there's really three parts to the cytoskeleton, three bits. Okay? But roles, support, number one, right? You saw from that picture that it gives the cell its shape. Right? The two microtubules push out, and also the actin filaments, the microfilaments, attach to cells, allow for attachment, and they allow for the cell to have shape and structure. Okay? Cells move. Do you agree? Yes? I mean, even cells that are attached to something, a plate, right, in an incubator, it still moves, right? You get a cut. Eventually, those cells are going to migrate and are going to heal that wound. Yes? Okay. Well, how does that happen? How does a cell move if it has this cytoskeleton? What does this cytoskeleton have to be? Flexible. It has to be dynamic. Okay? It's not stagnant. It's not just built and stay there. Okay? It is built and it can be broken down in different parts. Right? So if I wanted the cell to move, a certain way, if I had my cell here, and I want it to move this way so that it's eventually going to go like this, right? And then, it's going to look like that. Say the red being the side of the cell that's moving towards, right? So red being eventually, let's draw that a different way. Okay, let's say. So this is your original cell, okay? And this cell is going to move, okay? So that this edge is going to move this way, okay? So eventually you're going to end up at this red part, this front, okay? So that's the cell front, the edge of the cell, okay? So how is that cell going to go from the black line here to here? Does it have to push out? is the membrane just going to go on its own? Or how are we going to do it, Brett?
Like an anchor and a pull system?
There is a little bit of an anchor and a pull system, right? And there's also a pushing system. So we're going to push the membrane out after we make an anchor, right? So those two parts that we just talked about, that microtubules in green, are going to push out the membrane after the red spots, the microfilaments, produce an anchor and pull it. Motility. Okay? How cells move is dependent upon this. How things within the cell move also depend upon these. Okay? So if I have a cell, here's our cell, right? Nucleus, endoplasmic reticulum. We know the ER, we'll call this the rough ER. Right? What does the rough ER do? Produces what? Proteins. Okay? So now it produces a protein, it produces a vesicle with a bunch of proteins inside. And I want those proteins eventually to get out here. How does this vesicle get to the membrane? Does it just eventually get there? Floating around, happy as can be, it'll get there eventually. A bubble in the air that's floating in the wind. That's not efficient. Okay? So if I had a track that this vesicle could follow, that would kind of pull it along, eventually get to the membrane, and allow the protein out. That would be much faster. You agree? Yes. And that's what the cell does. Okay? So it utilizes the cytoskeleton as a highway to pull things along. All right? And it does this through molecular motors, such as dynein. Okay? So here's a microtubule. Right? You see this is in green, the microtubule, right? That's an element of the cytoskeleton. Here's a vesicle, a couple vesicles, that are attached to that microtubule, and this molecular motor, there's a motor on there that's going to move along the microtubule and stepwise pull it along until it goes where it needs to go. Okay? So motor proteins that actually walk along, hydrolyzing ATP and pull them along. So motility not only for the whole cell, but within the cell as well. So moving parts along. Questions on the functions of the cytoskeleton. So support, shape, right, but also movement structure. Yes?
So microtubules push out the cell, and then the microfilaments, like they're structured in like a circular, right?
Yes. You'll see it. When we go through the components, you'll see it a little bit better.
Okay.
Okay.
I was just going to ask, like, what the microfilaments, like, how it helps with the structure. Does it just keep it, like, the same circular shape?
They allow for contact points. All right. So if I was to look,
Within the cell.
So most of the time, I've been drawing cells like this, right? And that's looking from the top down through the cell. What if we look from the side? The cell was like this, right? And here's our nucleus. We're looking basically like this from the side. Yeah. Okay? So here's our plate. Say this cell is growing on a plate. That's our plate, right? So now the cell isn't just kind of floating there, right? Mammalian cells, animal cells, have to be attached to something in order to grow, right? And so your attachment points are actually these little points on here, right? So if I was to draw them along, they're kind of going like this. And you see them underneath the cell. But as you're looking through, it looks like they're within the cell, but they're actually underneath. If you were to look to the side, you see that they are underneath. But then that's near impossible to do. You can't really do that, right? So, but the understanding is that these are attachment points for that. So these anchoring points are the microfilaments, right? And then within the cell, we have our microtubules that push out, right? And it kind of looks like this from here, right? So they will push out, they'll push out the membrane as it needs to, and then as it pushes out, this part, say this cell's going to go this way, right? We'll have to make a new attachment point and then break up old attachment points to allow the cell to move in a certain way.
So those are outside of the cell?
They are, yeah, they are outside and within, right? So they're both. 3 parts, right? We have microtubules, microfilaments, and intermediate filaments. Microtubules, okay, are the largest part. They are the largest, thickest cytoskeletal element. Okay? So even though it says microtubules, it is the biggest. All right? The next are intermediate filaments. All right? They're kind of in between. And then you have your microfilaments, which are also called actin filaments, and they are the smallest. Okay? So in order of size, all right, from smallest to biggest, microfilaments, intermediate filaments, and microtubules. Right? That can be somewhat confusing because of the microtubule. Right? Micro does not mean, in this case, the smallest. This table is very important. It gives you everything that you really need. Okay? The structure of microtubules is a hollow tube. That's why it's a tubule. It's a hollow tube composed of tubulin subunits. So tubulin's a protein. There's alpha and beta ones. And they link together to form this helical tube. That's hollow. Okay? They function in maintenance of cell shape, cell motility, especially in cilia and flagella. Right? We know what cilia do. We know what flagellum do, right? Microtubules make up the structure of those two modal structures, cilia and flagellum. All right? And so it also has to do with organelle movements within the cell. And very important, during mitosis, during cell division, microtubules actually pull chromosomes apart. Right? They actually pull them apart during cell division. Okay? Excuse me. Gosh. Microfilaments, actin filaments. Okay? They are smaller, and you see the diameter of them. They're composed of actin, right? And actin in a helical nature. So we're going to add subunits here. Why is it important that these are adding subunits? So if I can add subunits, what else can I do? I can take them away. What is that important for? If the cell's going to move, how am I going to push that cell membrane out? By adding subunits. Okay, I continue to add subunits, the membrane is going to be pushed, right? I'm making it longer. Okay? And if I have to break down other parts, I can take the subunits away. Right? Dynamic. This is dynamic. They can be put together and broken apart. Right? They can be changed. Okay? The intermediate filaments are less dynamic. Right? They're a little more stagnant. You see, they form the nuclear lamina. What's the nuclear lamina? So the nucleus has a shape, right? You look in a microscope, you look at cells in a microscope, what's the nucleus look like?
Sphere.
A sphere. It's round, it's circular, right? It's pretty much that shape all the time, right? How? Why? Does it have to have something in it that holds it that shape? Yes. These intermediate filaments give this lamina, it's a structure to the shape of the nucleus. All right? It's like a netting. right, that gives it structure. Helps maintain cell shape as well. And really a big one, right, the anchorage of nucleus and certain other organelles to the cell. All right, so now we can go through them all. Each is basically going through each of the next couple slides, go through each of the bits, the three components of the cytoskeleton like we just did. All right, I gave you the table. We went through it. All right, so we can go through it pretty quickly. All right, in terms of microtubules, there's a point where they organize and they radiate from. Radiate, what does that mean? Here's the center, radiate, it means kind of come out in all directions, right? Those are arrows, poorly drawn arrows. Okay, so there are a couple different structures that you need to know. Centrosomes and centrioles. Okay, in animal cells, microtubules arise from a microtubule organizing center called the centrosome. Right, so here's your centrosome. The microtubules are going to radiate out from that point. Right, so there's a starting point, and we know which direction to go. Okay, the centrosome actually has a pair of cylindrical centrioles. Okay, each with 9 triplets. So 9, 3 rings. So 3 rings times 9. Okay, in a center, in a circle. All right, that's what a centriole has. Each centriole has 9 triplets of microtubules. So it's a collection point where all these microtubules come into. All right, what does it look like? It looks like this. Okay, here are your 9 triplets. Okay, you're actually showing plasma membrane portion of a microtubule doublet. Okay, this is cilia and flagella. We'll talk about them in a second. All right, here's your 9 triplets. Centrioles, 1, 2, 3, 4, 5, 6, 7, 8, 9. Triplets, and they are arranged perpendicular to one another. Okay, they're usually somewhere around one side of the nucleus. All right, and it is an organizing center. This is where the microtubule cytoskeleton will originate from. Okay? Questions on centrioles. 2 centrioles make what? 1 centrosome. Okay? And all animals have it. All right, our last thing we'll talk about today are cilia and flagella. So microtubules are important for the movement of these 2 structures. All right, they are composed of 9 doublets, not triplets, 9 doublets, and plus 2 more inside. So you have, so 2, that's a doublet, and we have 9 of them in a ring. And then in the middle, we have 2 more. Okay, that's the structure of a cilia or a flagella. All right, and what do cilia and flagella do? So they beat, right? They move back and forth. All they do is just kind of this motion. Left to right, or up and down, up and down. Okay? The structure of the microtubules allow that to happen. So, where are cilia and flagella usually in the cell? Are they in here? They're on the outside, right? So flagellum's a long extension, and cilia are shorter extensions, but they are outside of the plasma membrane. So there is part of it that actually is encompassed and enclosed by the plasma membrane. All right? There's an anchoring point where the microtubules anchor or attach to the cytoskeleton of the cell. all right, and your motor protein. When I say motor protein, what do you think? There's movement, okay? So this motor protein dynein is actually going to change shape and cause the movement of the cilia and the flagella. All right, let's structure. So here's the structure. Here's your 9 doublets, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. Plus the 2 in the middle. That's your central microtubule. You see here, right, they are attached to the central microtubule by other proteins. So your doublets are attached, and then you have these other proteins that attach the doublets to each other. Those protein, the attachment is important for the movement. Okay, you break that motor protein is going to change shape and cause the movement of the cilia and the flagella one way or the other. Okay, then your basal body. This is the anchoring point right here. What does that look like? It looks like the centriole. Okay? So the extension, the cilia and the flagellum look different than the basal body. The basal body looks more like the organizing center, the attachment. All right? And then it goes through how the walking, the movement of dynein move things around. We'll stop there for today. Last class, we covered the cytoskeleton. Okay? So we covered aspects of the cytoskeleton that give the cell internal support and also a mechanism of internal motility and cellular motility in general. Okay? Any questions on the cytoskeleton? You know the 3 elements, the subunits that make up the 3 different elements of the cytoskeleton. You should have an idea about them. Okay? But now the next part is actually going outside of the cell, okay? Because the cell is going to make contact. All mammalian cells, and you can write this down, all mammalian cells, okay, have to be contacted, right, or what we call adherent, attached to something, in order for them to grow and function. Okay, there are some cells that float around on their own, okay, but like red blood cells. But guess what? They're not growing. Those cells, red blood cells, are not actively growing. Okay? So cells need to be attached to something. What they attach to is what we call the extracellular matrix. Okay? And most cells, so in the lab I can take a group of cells, so cells that are floating in solution. Right? So if I have a little test tube here, and I have all these little cells in there, and then I look at it like this, say one of those like that, and there's all these cells floating around. They're in solution, and they're not adherent, okay? But then I put them on a plate, a plastic plate, and then these cells will start to, is anyone red-green colorblind? Just making sure, right? So you can see this, right? Those cells will start to, by gravity, they'll go down to the bottom. All right, and after a certain period of time, what's going to end up happening is these cells are going to secrete proteins and stuff and produce junk outside of the cell that they would start to attach to. They actually make their own anchoring points. All right, they make their own extracellular matrix. Or I could be nice and I could put the proteins on the plate already and it would make them attach faster. Okay? But the extracellular matrix, the proteins that are on the outside over here, right? Outside of the cell, over here. That's important for how the cell recognizes where it is, what its context is, what's surrounding it, etc. Okay? You'll know I did not, how did I draw those cells, those red cells? Are they bunched up next to one another, or are they spread out a little bit? They're spread out, okay? Because on a plate, what's going to end up happening is, these cells will start to divide. So this cell will divide into two, and then it'll make a new cell here, a new cell there, and that cell will divide into two, and it'll start. What ends up happening? Kind of fills in the space, right? Yes? So at some point, the cell's going to recognize that there's no more space. There's no more room to grow. Okay? And so the extracellular matrix, there's no more space, there's no more extracellular matrix. That's one signal for the cells to stop growing. No more space, stop growing. No more proteins to attach to, hold on. Okay? So that's what we call density-dependent inhibition. Too many cells in one space, cells stop growing. Okay? But let's talk about some aspects of extracellular components, extracellular matrix. One of the biggest ones is cell walls of plants. All right, we know what cell walls are. We're familiar with them, yes? Yes? Okay. All right, it is an extracellular structure. So if I was to draw the cell wall, say this is the cell wall, the plasma membrane is interior to that cell wall, right? The plasma membrane is in black, the cell wall is in green. So in all essence, every plant cell is separated from another cell by its cell wall and also its plasma membrane. The cell wall in plants is mainly made of cellulose, a polysaccharide. Okay? It helps for protection. It helps for protection. What it does, it helps protect the cell and prevent it from bursting when the plants are immersed in water. Okay? How many of you heard of the word hydroponics? Anyone? What's hydroponics?
When you grow it like with water with nutrients and not soil.
So we're growing plants in water, period, with and then some added nutrients that plants need, but no soil. Okay? It is what you put a plant in pure water, what should happen? All right, what's going to happen in terms of osmosis? More water on the outside, water's going to rush in. If you just had cells, what's going to happen? Those plant cells are going to burst. Why doesn't that happen in plants? The cell wall. Okay? So water, right, rushes in. High water on the outside would rush in, but that only makes the plasma membrane push out. It's still contained by the cell wall. Okay? So, in the summertime, how many of you at home have gardens or parents have gardens? In the summertime, it gets pretty hot, right? In the afternoon, after a hot day, the leaves start to kind of droop, right? They start to droop because they're wilting. Lack of water. You water them, you throw the hose on for half an hour, whatever. What happens? They uptake water, and those plants, they're going to no longer wilt, right? Because what's happening is water is rushing in these cells and pushing back on the cell wall and making the cells, the plant cells, what we call turgid. Okay? They are pushing out and actually pushing out against the cell. Okay? Plant cell walls have multiple layers. There's a primary wall, a secondary wall in some cells, and there's this thing called a middle lamella, right, which is a thin layer. In terms of structure, it doesn't really mean anything, like the structure doesn't really change and doesn't really change its function because we know what cell walls do. They provide protection, okay, and then some structure for the plant. But just understanding that it is built in stages. All right? But the next part is important. Cell walls have, now one cell in a plant is not completely separate and functioning on its own from the other cells around it, even though it is surrounded and cut off by a wall. Say, this class versus the class next door, right? Now, if we wanted to have some communication with that class, I could open the door, right? In effect, that's what a plasmodesmata is. It's a hole in the wall, in the cell wall, that allows all the cells to communicate with each other. So the doorway is analogous to a plasmodesmata. All right? It's an opening between the cell wall that allows all the cells to communicate. All right, so here's what it looks like. Here's your primary and secondary cell walls, right? There's a middle. Why is there a primary and secondary cell wall? What's the middle lamella actually? If I had two plant cells, here's a plant cell, here's a plant cell. Where did the cell wall come from? Who builds it? Not Donald Trump. Right? Who builds it? Each cell is going to build its own, right? So now this cell is going to build its cell wall, and this cell is going to build its cell wall. That's what you're ending up with right here. Here's one cell wall, here's another one. They push up to one another, and you end up with this middle part in between. That's what the middle lamella is, okay? And then what we have down here is your plasmodesmata. Okay, you see the channels that run through the cell wall and allows connectivity, right? It allows communication between one cell and the other and this cell. So now all these cells are now going to function as a unit as opposed to you do one thing, I do another. Okay? Questions on plasmodesmata? Think about that. Remember the name, plasmodesmata. Okay? Oh, no, I want to add one. We're good. Okay? Questions on extracellular components, the main extracellular component of plant cells. Okay? And it's mainly a polysaccharide. All right, there are some proteins as well in the cell wall. In animal cells now, we have what's called the extracellular matrix. It is not a cell wall, but it is proteins and polysaccharides and what we call proteoglycans. All right, proteo refers to protein, glycans is sugar. All right, so we have proteins and sugar that are connected. And then this other protein called fibronectin. All right? These proteins are produced by the cell, and then the cell actually has like little receptors. All right, so here's a receptor here, and they function in heterodimers. These receptors are called integrins. Okay? And these little receptors, they have binding sites for the proteins. Okay, so then they attach, and now, what does that do? So I have this receptor on the plasma membrane here. I have a receptor on the plasma membrane that's going to attach to this protein in, so here's a protein. I would just put it as an X right here, right? So in green up there is the extracellular matrix. Now the cell's going to attach to this protein. What's important that it's in the membrane of the cell? What allows that, what does the positioning of the integrins allow for the cell to do? What do you think? How does the cell know it's attached to the the extracellular matrix? We just said it. We just had what's called adherent dependent growth, right? We have to have the animal cells have to be attached to something in order to grow. Yes? So what do you think this is? Is this one mechanism by which the cell can say, yes, I'm attached, I can grow, or no, I'm not, and to cut off? Exactly. Right? So these integrins attach, and they send a signal into the cell that says, it allows the cells to start growing. Sam?
Can you repeat what the relationship was?
Adherent dependent growth. All right, adherent dependent growth. Can you read that, chicken scratch? Okay? This is one mechanism by this happening. Okay? So these receptors attached to the proteins, they send a signal inside, that sends another signal, and then that turns genes on to have the cells start growing. Okay? Pretty straightforward, yes? But without that attachment, cells don't grow. Okay, so this is kind of what it looks like. All right? So you have, these are your integrins. Here's your alpha, right, and beta subunits, the two subunits. You don't have to know alpha and beta. Just know that they are always, there's always two parts to this receptor. So it's attached to fibronectin, right, this fibronectin protein. There's some polysaccharides. The importance of the polysaccharides is that it helps retain water. Okay? These span the membrane, and then they are attached. They have some type of attachment to other proteins inside. And not only could it be the signaling to say the cells can start growing, but it also could be structural. They form anchoring points. Right? So it's going to anchor to the extracellular matrix, and now that cell's not going to move anywhere. It has something to attach to. Right? It's something to pull on, push on, whatever it might be. So the cytoskeleton actually comes into play here too. So microfilaments, which are made of what protein? Actin. Okay? Actin. All right? Questions on that? So what if I were to say I gave you a pharmaceutical agent, a drug, that disrupted microfilaments? They somehow caused microfilaments to depolarize and break up. What would that do to cellular growth, do you think?
Stunted.
Why? Why would it stunt it?
Because there would be no attachment points or no recognition of.
There's no recognition of attachment points. Okay, so this whole complex here, microfilaments, the integrins, the extracellular matrix, that's the signaling to say we can grow. So if I break this up, that signaling can be altered. If I block the integrins by somehow, I change the way they are, but okay, and they're not going to attach to the extracellular matrix, no growth. Okay? If I change the signaling so that the cell thinks that these integrins are always attached, even though they're not, then what's going to happen? What if there's a gene mutation where the integrins are always sending the signal to the cell that we're attached, we are good? They do what? They keep growing. So now they keep growing. Even though they're in solution or they're not attached to something, they keep growing. Then they float around, they go somewhere else. And then they attach, and then they keep growing. What's that? Cancer. It's metastasis. How many of you have heard of metastasis before? Okay? The cells let go of the extracellular matrix anchoring point, but they're still getting the signal that they're there, and they're okay. They grow in solution. They grow in out of their normal spot. They go to another spot, they attach, and then they start to grow again. That's another tumor. Okay? That's a big problem. Okay? So, example, the application of that. All right? Questions on extracellular matrix? Okay? Last thing we need to talk about are connections between cells. We already talked about one of the connections, plasmodesmata, which goes connecting plant cells between cell walls. Okay? There are 3 other types of cellular junctions that are important, especially in animal cells. And the reason why we talk about cellular junctions is because in biological organization, what do groups of cells of similar structure and function do? What do they become? What's the next step up in our tissues? Okay? So if I can link a bunch of cells together that are similar in structure and they do the same thing, well, then they could have even a bigger function. They can contract. One cell contracts, but lots of cells contract, and now that's a muscle, right? And that muscle moves something, okay? So these junctions are important for that purpose, okay? 3 types. We have tight junctions, which are linking of the plasma membranes between cells. Okay, we're going to link plasma membranes. We're going to have desmosomes, which are like buttoning a cell to something else or riveting it. Like it's an anchor point. We're going to actually attach part of the cytoskeleton between cells. Okay, and then we have gap junctions, which are little tunnels that between 2 cells that allow them to communicate to one another. Similar to a plasmodesmata. Okay? So we went over plasmodesmata, right? What runs through? They perforate cell walls. Water can go through. Salt, some small solutes, maybe some RNA or some protein can pass from cell to cell. It allows for communication between 2 cells that are next to one another. So they're not completely on an island, right? They're not completely separated or segregated from each other. Now your tight junctions, all right, 3 main, we have tight desmosomes and gap junctions. These are in animal cells, and this is the important part. They are all found. They are extremely common in epithelial tissue, okay? Epithelial tissue is the tissue in your bodies that cover things. They provide a barrier between outside and inside. All right, your skin. Think about your skin as a prototype or your stereotypical epithelial tissue. Protects you from the outside versus inside, keeps water in, keeps, all the other stuff you have inside your body and everything else that you don't want out. Okay? So let's work. Okay? Tight junctions are up here at the top. Okay? I'm going to draw them out in a minute. But what you see here is the tight junctions link the plasma membrane between the 2. Okay? So 2 cells, I'll draw it out in a second, but the 2 cells are actually zippered together. So now nothing can get in between the cells, effectively making the cells a barrier. How many of you ever played Red Rover when you were a kid? Okay? So the way you play Red Rover, right, you link arms and you say, send someone else from the other side over. When they run through your arms, what are they doing? Your arms, your hands or arms that are linked are analogous to this tight junction. You're preventing anything from getting between your line. Okay? That's it. That's what a tight junction is. The 2 cells are married to one another in such a way that nothing gets in between them. So now they are an effective barrier. Okay? The desmosomes are right here. That's going to link the cytoskeleton. Okay? Providing a stronger component. So now these two cells don't separate from one another. Now they're a functional sheet. Okay? The tight junctions are good for preventing stuff from getting through, but not so good in that they aren't strong enough to keep cells from peeling apart from one another. Okay? And then you have your gap junctions, which are little tunnels. Okay? And they allow for stuff to go through. I'm going to go through them again. I'm going to draw them out. Okay? So here's our cells. Let's do the cells in blue. Here's cell 1. Here's cell 2. Right now, cell 1 is completely separate from cell 2. All right, so now we're going to put in a tight junction, and this cell membrane of cell 1 and cell 2 are going to be married to one another. So now we're going to actually, let's move this thing a little closer. Now, if this was three-dimensional, what we're going to end up with, we're going to form a little dot there, which extends out. Nothing gets in between. So literally, the cell membrane pinches in. Okay? So now nothing can get between those two cells. Okay? Questions on tight junctions? It's like you're zippering that part together. The desmosomes have your cytoskeletal elements that are then linked together, bundled, and there's proteins that connect between these two. So now you have two proteins, you have a group of proteins that link to one another, and then this ends up being what they're linked to is the whole structural element of both cells. You end up with the two cells not being able to be pulled apart. Okay? So the tight junctions are a functional attachment that doesn't let anything in between. The desmosomes are a structural attachment that doesn't let the cells split apart. All right? So one's functional, one is structural. Excuse me. And then finally we have our gap junctions. Okay? Our gap junctions are a tunnel between the two cells. Okay? A tunnel between the two cells so that whatever's made over here, well, let's do it in black. So if I have something here, it could go through the tunnel and come out over here. Allows for communication between the cells. So in order to if I were to break you up into groups, into Red Rover teams, okay, right? You're going to have elements of all three of these in order to function as a good Red Rover team, right? You have to have some tight junctions, right? So you have a barrier, a little bit of strength, so maybe it's not just holding hands, it's more like locking arms, and then you have to have communication, right? You have to know where your stuff is going between one another, or if I'm not pulling each other out, all right? So this is how cells use these connections in order to function as a good unit. Okay? Questions on that? Now, we've been talking about the cell and the organelles of the cell. Understand that the cell's ability to do stuff is a sum. It's greater than the sum of all its parts. Okay? That all the parts of the cell, they do stuff, but it provides function, right, for the cell to do lots of different things. Right, by coordinating efforts of the cytoskeleton, the plasma membrane, etc. And your book uses a macrophage as an example. All right, a macrophage is a cell that is, you have them flowing throughout your body all the time. All right, they're found in the blood. And they travel around the blood by kind of rolling along blood vessels. Rolling along, right, happy as can be. You're in good health, no problem. Well, at some point, they recognize that there's a cut or an infection or something. Say you get a cut and there's bacteria that are introduced into that cut. The bacteria cause swelling and pain, the bleeding or the cut blood vessels. They send signals to the macrophage. It's like a little beacon, a lighthouse. Woo-hoo, something's wrong. We need you guys, okay? The macrophage recognize the signal. They leave the blood vessel. They squeeze through. How do they squeeze through the blood vessel and move to where it is? Cell motility. That requires signals to be interpreted, received and interpreted. That requires cell motility. The cytoskeleton has to be dynamic, has to be reorganized in order for things to move. That includes the cell membrane, has to be able to be reorganized and moved. That includes lots of different genes that need to be activated. All of that's happening between lots of different things. Then the macrophage gets to the point and it recognizes a bacteria, it grows around it and engulfs the bacteria into a lysosome, into a vacuole first. Then the lysosome within the cell fuses with that vesicle and causes that bacteria to be digested, taking it out of circulation and removing it from the body. Right? All of that is important. Now, I told you about the chart at the end of the chapter. Okay, that's important for your structures. And this is not a complete chart. Right? You have your nucleus. You should look at that. You also have the different parts of the endomembrane system that you were responsible for. Okay, so not only are your study guides that you posted for homework, but also for this chart, it kind of puts it into a small section. Then you have your mitochondria, chloroplasts, peroxisomes, and also this continues with cytoskeleton, etc. Questions on these parts, the workings of the cell. Okay? On Monday, we're going to go through we're going to start cellular functions. I want to talk about active transport, passive transport, osmosis. My goal is to get all of those three things done on Monday. That's an ambitious goal. Okay? But I think you guys can do it. All right? So in order to do that, please, over the weekend, go through, listen to the lectures from last year on active and passive transport. I will post them today, okay, as I post this one as well. Questions?